Welcome, science friends, to Mendel's laws of heredity. So we're going to go back just a little bit and remember Mendel. He was the sort of grandfather of modern genetics. He did all the pea plant experiments. He went and investigated genetics without knowing all of the stuff we know now. But we can sort of work backwards and apply what we're learning now to what Mendel was discovering without all of this common current knowledge. So first I want to speak about Mendel's law of segregation. So during meiosis, alleles segregate. Those homologous chromosomes are separating, and each allele for each trait is packaged into a separate gamete, which is how we get those 4 haploid gametes at the end of meiosis. We're seeing all of those different alleles separate out. We see this happening, and Mendel didn't know about meiosis yet. But with his investigations, he knew that something was happening inside of these gametes where we were seeing these different alleles separate. So over here we can see, looking at just the alleles and the letters we use for alleles, if there were 2 alleles, one from the mother parent plant and one from the father parent plant, each offspring gets one of those alleles from mom and one of those alleles from dad. It doesn't get both alleles from mom and both alleles from dad. So he knew there was something happening in this process of sexual reproduction where only one of the 2 alleles, or half of the genetic information, was coming from the mother plant and half was coming from the father plant. And we see that now with the knowledge of meiosis, that yes, that separation happens during the process of meiosis, but Mendel was able to figure this out without knowing about meiosis. It's kind of wild and crazy. We see this law of segregation when it comes in the meiosis process. So here we have 2 homologous pairs of double-stranded chromosomes, or the X shapes. And we're going to separate those 2 homologous pairs during meiosis 1. And then during meiosis 2, they get split into halves to create our 4 haploid cells. Again, one allele goes in each of those haploid cells. Just like we see with the alleles and letters, one letter, so one allele of that total gene. You need 2 alleles, one from mom, one from dad, to create that zygote or that offspring. The other law is called Mendel's law of independent assortment. We see independent assortment in meiosis as well. Again, Mendel didn't know about meiosis yet, but he was still able to come to all of these conclusions without that knowledge. We see different genes separate into the gametes independently. So it's not like, oh, I see the gene over there for eye color and I'm the gene for nose shape, I need to be with them. No. DNA chromosomes do not have brains. They don't need to be with other alleles and other genes and other chromosomes. They align themselves differently. It's all random. We see non-homologous chromosomes align independently. Those classes of gametes are produced in equal amounts. There's going to be one allele for hair color, one allele for jaw shape, one allele for height. All of these things are separated during the law of segregation, and they are independently assorted during metaphase 1. This is true for genes on separate chromosomes, or it's also true for the same chromosome, but only if crossing over happens frequently. So let's look at how the law of independent assortment relates to meiosis. We see independent assortment happening during meiosis. We see it happening during metaphase 1. The only exception to this law are if the genes are on the same chromosome or are close together. So usually if they're close together, if they cross over, they will be crossed over together. Or if they're on a smaller chromosome that doesn't have a lot of crossing over that happens, usually they'll be inherited together. When genes are rarely crossed over separately, they are considered what we call linked, or they're almost always going to be together during the meiosis process and therefore inherited together, and they are linked, so to speak, because you always see them together. So here's an example of that independent assortment. We have 2 possible arrangements during metaphase 1, which will lead to different arrangements in the gametes. So how those homologous pairs align along the metaphase plate can change the gametes and mix all those things up, providing genetic diversity. Really quickly, here's a Mendel's law recap. We have the law of segregation, where we're looking at single traits, and those alleles are separating out into the gametes during anaphase 1, and therefore also during anaphase 2. How they get separated, we're just saying one allele goes to each of those different gametes. So if we have big P, We get one big P going to one gamete, the other big P going to the second gamete. So it's not two of those alleles going to the same gamete, they are separating or segregating from each other. The other one, the law of independent assortment, we see genes on separate chromosomes assorting into gametes independently. Again, DNA and chromosomes do not have brains. They can't say, oh, well, the gene for eye color is over here, so that means I have to move over here or I have to go with that one. No. It's all independent. They all get sorted into the gametes evenly, just randomly. This is happening during metaphase 1. And what happens in metaphase 1 dictates what happens in metaphase 2. The only exception to the law of independent assortment are linked genes. Again, these are genes that are very, very close together and are very rarely crossed over without the other. And so we see them being inherited together very, very often. That was Mendel's laws of inheritance. I hope it helped.